Our speaker for today is Professor Romain Quidant. Professor Quidant received a PhD in physics in 2002 from the University of Dijon, France. He then joined the newly created ICFO in Barcelona as a postdoctoral researcher. In 2006, he was appointed junior professor in tenure track and group leader of the Plasmon Nano-Optics group at ICFO. In 2009, he became tenured professor both at ICFO and ICREA. After nearly 18 years at ICFO in June 2020, he joined the Mechanical and Process Engineering Department, DMAVT, at ETH Zürich. He is recipient of 3 ERC grants and several international and national prizes. Since January 2022, he serves as the editor-in-chief of the ACS journal, ACS Photonics. Thank you so much, Professor Quidant, and the stage is all yours now.
Thank you very much, Neha, for this very nice introduction. It is a great pleasure to be here today and to participate to this Science Talks. So let me share the slides. Can you see them?
Yes.
Very good. So great pleasure indeed to be here. So considering that we have a quite broad audience, I'm sure, I decided to talk today and to share with you today a quite interdisciplinary aspect of our work, and where we're going to use a quite actually mature optical technique, so-called digital holography, to revisit the field of biosensing. So because I don't want to rush through that at the end when I will not have time, I want to first acknowledge my team. So I feel very grateful to work with a group of very smart people from all over the world. And I would like to particularly thank and acknowledge the work of the people that have participated to the science I'm going to present today. So Alexia Stolman, Rebecca Büchner, 2 PhD students in the group, Andrea Sottini, a postdoc, Jose Garcia Guerrero, who is actually our expert in nanofabrication and microfluidics. And finally, Jaime Ortega Arroyo, who is a senior scientist in our lab and who is coordinating, let's say, a big part of our bio-nanophotonics activities. So we're going to talk today about, as I said, a technique that is actually quite mature. This is what is called digital holography. So digital holography is kind of the modern evolution of what of the technique that you see Denis Gabor on this black and white picture presenting on the blackboard just after receiving the Nobel Prize in 1971 for the invention of holography. So in a typical digital holography setup, what you would have when you look at the figure on the left, you would have a light source that would be splitted in 2 paths. There would be first the object path that would go through, that would interact with the object and carry some information about it. So in that case, the light is actually transmitted through the sample. And on the other hand, you would have what is called the reference beam. So this is the other one we see on the right. And so these 2 beams will, at some stage, recombine with each other onto a digital optical sensor. So in that case, a CMOS camera. And so they could recombine with, let's say, fully aligned with an angle theta or equal to 0. In that case, we talk about inline holography. And that can be recombined with an angle different from 0. In that case, we talk about offline holography. But what is very powerful about this, in fact, quite simple approach is that on the sensor, you're going to form an hologram, and this hologram will enable you to extract both the amplitude and the phase of the electromagnetic field. So this is a technique that has been implemented in many different forms and has contributed to many different scientific and technological directions. for instance, for label-free microscopy, for tomography in medical imaging, cell identification, classification, material sciences, and metrology and inspection. So what I would like to do today is to try to convince you that this technique could actually be extremely relevant to address some of the major challenges of today's, let's say, modern biosensing techniques. So in particular, we'll focus on two main topics. So the first one on the left is how to use digital holography to do molecular fingerprinting of extracellular vesicles that are actually secreted by cells. And in the second part, I would like to show that we can actually go a step further and extract information and map optical activity, so structural, basically chirality of matter using a digital holographic microscope. So let me start with the first topic. And I would like to spend some time to motivate and to give a little bit the long-term picture behind this technology. So I'm sure you know that there is a very big trend lately in developing the so-called organ on chips. And so these organ on chips are actually somehow hosting, so integrating grown human tissues into a microfluidic environment. And the idea is that two main motivations behind that. The first one is the possibility of using these artificial organs in order to reduce the use of animal models and directly somehow experimenting on this artificial, let's say, platform. And the second one is actually because these organ on chip actually using human tissues or human cells, the idea is that basically the biology and the physiology of humans actually sometimes even better reproduced than it would be, for instance, in a murine model. So you can think about all kinds of applications. Of course, you aim at developing human-relevant disease models. You want, in the case of the pharmaceutical industry, to develop new drugs. And also going toward, so this beautiful idea of going toward personalized medicines, where the idea is that an organ or different organs on chip would be, let's say, put together for a given patient. And the idea is that this would serve to actually develop a given treatment to a disease of the patient, so toward this personalized medicine. So these are beautiful developments, but there are still many challenges. And these challenges are coming from the fact that we are talking about a very interdisciplinary kind of technology. And so the first complexity is actually how to reproduce in this artificial device all the complexity of biological system. And so one of the major, for instance, difficulties, how do you implement vascularization in such an organ on a chip? There are things that are more related to engineering and microfabrication. So how do I scale up? How do reproducible, let's say, organs, but also how I can interface what is more man-made, like the microfluidics, with the actual biology. And there's a third aspect that is related to the fact that if you really want eventually that organ on a chip to serve, let's say, the purpose as I defined before, what you need to do is to be able to extract like a large amount of information from them and to be able to use this information. So what I'm going to present in this first part of the talk is actually addressing this third point and illustrating that there is a need right now for technologies that are capable of doing real-time and non-invasive monitoring of the secretion of this organ on chip in order to kind of learn what is happening within the organ. So why is that important to actually get or to extract or to detect, better said, secretion from an organ on chip or from the cells that are contained within the organs, better said? So there are many reasons. The first one is that basically secretions are what makes that the cells are talking to each other. So it's very beautifully illustrated on this sketch you see on the right. That's a way, by monitoring the cell secretion, in fact, to provide some insight on the fundamental aspects of everything that is tissue maintenance and diseases. We should not forget that many of the biomarkers that typically we would routinely detect in body fluids like blood, they are coming originally from the secretion of the cells. So of course, these secretions, they contain a lot of information related to the detection or the monitoring of a disease. And finally, there are even right now research directions where the idea is to use these secretions to actually develop some new treatments. So what we aim at doing within the group is to develop some new platforms, optofluidic platforms that are capable of detecting some specific analytes within the secretions of these organs. And so we want not only to be able to monitor a few specific analytes within a complex matrix that is formed by the secretion, we want also to be able to do that real time in order to really be able to probe, let's say, the status of the organ to any kind of external stimulus or drug injection or stress that would be applied to the organ. So the complexity comes from the fact that actually, so of course, it's less complex than maybe a body fluid like blood, but still the secretion from these organs are complex matrices. And so they contain actually a variety of different molecules and biological particles. So for instance, hormones, proteins, protein aggregates, and extracellular vesicles. And all these different molecules, they have different sizes, and so they make basically the sensing step complex. So the first thing, so if you have this objective that I've just defined, so the first thing you may want to do is actually looking around at different sensing techniques and to try to use one of them in order to see whether I can actually serve the purpose. No, I can indeed tackle the problem. It's actually what we did originally, and so we've been working many years on this LSPR sensing chips. So what you see in between these two fingers, a microfluidic chip, and the more like bright spot you see in the center, actually arrays of gold nanoparticles. Typically, the arrays are 100 micron big, and there are many, many particles that are regularly arranged. And so what we do is that we, in that case, functionalize the surface with a capture molecule, for instance, an antibody, and we, upon binding of the analyte, we monitor how the color, the resonance of the plasmon and the color of the sensor is actually changing. And this information, this color change, gives us direct information about the amount of analyte. So what we did originally, and that was before moving to ETH, we were still in Barcelona, and so we initiated this collaboration with Javier Ramon Ascon from IBEC. And so what we did is he's developing in his lab some very nice organoship models for studying diabetes. And so in particular, in that case, we connected a chip to one of these pancreatic islets that he's developing in his lab. And the idea was to see whether we could directly, using this kind of sensor, monitor the secretion of insulin from these islets and see how this can vary basically with the concentration of glucose. And so this is actually what we did back then. And so what you see here is the change in the color of the sensor. So this is in nanometer, what is called LSPR shift. And so as a function of time, and you see these two different, let's say, parts of the graph where we have increased basically from 2.8 to 16.7, the concentration of glucose, and we see that the insulin secretion actually following up. So it's very interesting, promising, but it's still very challenging because the problem with the sensors is that you have this array of 100 microns with many particles, and in fact, the signal you extract via the measurement of the color of the sensor is something that is integrated over the entire array. all these particles are measured at the same time. And so you don't see the detail of what is actually binding at the surface of the sensor. You just see a modification on the environment. And so you rely mostly on the chemistry to capture the molecules. There's limited throughput, and there's also, let's say, limited multiplexing. So you're only limited to a few analytes. So if I think about it, the things we would like to have ideally is to be able to, in order to deal with this heterogeneous population that are coming from the secretion of the cells, is to have a technique that's capable to do single particle sensitivity. So each of the binding events should be resolved, to have a specific interaction, to do kind of molecular fingerprinting. We want this information about the analyte size, knowing whether this is a single protein, an aggregate of them, whether this is an extracellular vesicle. And ideally, of course, we would like to have high throughput. So what we do in the lab is to combine digital holography, so that would be microscopy to detect single binding events, computer vision to deal with all these many single events that we're going to measure, some surface chemistry, because we still need some, let's say, biochemical specificity, and finally microfluidics, because microfluidics will enable to do many different experiments or assays on a chip. So I'd like to focus to advance on this project on a very specific type of biomarkers that are called extracellular vesicles. So vesicles, extracellular vesicles, are vesicles that are constantly secreted by cells, and they are among these vehicles that are used for the cells to communicate between each other. So in fact, you should see them so as a sketch on the top right. So this is a vesicle that has some very specific biomarkers at the surface, some what can be protein markers that are actually specific to the cell they come from. And in the inside, there's also some proteins and genetic materials. So what is complex about this kind of analyze that they're extremely heterogeneous, because the size ranges all the way from 30 nanometers to 10 microns. And so they are different subpopulations. It's actually a very complex analyte to detect, because actually there's no dedicated platform for that. And typically, the sensor I was showing before with the plasmons would not be able to actually discriminate what is happening at the surface. So I'd like to show a first platform using digital holography. In that case, this is an inline kind of digital holography, and this has been named originally IceCat by the two researchers who originally developed this this technique at the beginning of the years 2000, and that was actually here in ETH. So that was Veit Sandatkar and Philip Kukula. And so this IceCat technique is aimed at detecting small scatters that are just binding at the surface of a chip, of a cover slit. And so the whole idea is that you send light in reflection mode. So you focus in the back focal plane of the objective. This provides a collimated illumination on the scatters. And so the scattering of the actual object, if this is something like as small as 30 nanometers, this is very weak. What you're going to do is that you're going to make it interfere with the reflection at the interface with the, so between the glass and the upper medium. So this is basically the reference beam. So we have the object beam, reference beam, and eventually we form the hologram at the surface of the sensor. So eventually what you have, that would be the only formula that I'm showing today, is a signal for this IceCat. They're going to depend, of course, on the scattering of the reflected, on the reflected beam, the scattering from the particle, but most importantly on this interference term. And you can see that the very weak scattering is multiplied, is enhanced by the reflected beam. So this will boost it, enhance it. And the second thing that is important is that this scattering signal is proportional to the size, to the volume of the analyte. So that provides information about the dimension of the analyte. So eventually, this label-free, high sensitivity down to the single protein levels that we're going to show later on, and it's very well suited for heterogeneous population because you're able to resolve the size. So there's been a huge amount of progress in this ISCAT detection, particularly in terms of sensitivity. But if you really want to do something like practical, if you want to be able to do, for instance, a bioassay, you need to tick a few boxes. And the first one, obviously, is to have a proper chemistry. And so you need something, a chemistry specific to capture the analyte you're interested in. And you need also passivation, of course, to avoid non-specific binding. But this is more even critical because your ISCAT is so sensitive to whatever will bind at the surface. You need the surface to be ultra smooth, and you need also the surface functionalization to be compatible with microfluidics. So I want to show the approach. It's actually quite nice. We use some liposomes that have been doped with some biotin. And so we start from a clean glass surface. And we're going to use the same ISCAT microscope to see how the functionalization will happen real time. And so you're going to see all these black dots are going to appear on the surface. They are the liposomes, and they are binding on the surface, and they're going to start to rupture. And this is what will make that you're going to start to homogenize a little bit the surface, and you can monitor that in real time. But unfortunately, there are many of them that remain unruptured. So what you're going to do is that you're going to kind of favor rupture by adding a peptide, and eventually you're going to form an actual very smooth layer. So this is what you see here. So what is smooth? Well, smooth is what you see here. If I look at a channel that is 1 millimeter long and 200 micron wide, and I just extract randomly three different, let's say, zoom onto this larger area, what you see on the bottom, what you're going to see there's typically one defect. So one non-ruptured liposome over 200 micron squared. So it's a very clean surface. So we have managed this part, and then we need to check that the rest of the surface bioconjugation will not affect the quality of the surface. So we put first neutrovedin that attached to the biotin doping, and then the actual antibody. And so what you see on the right is a quantification of the roughness to check that, in fact, even by adding these additional steps, we don't degrade the quality of the surface. The second box to be ticked is beyond the surface chemistry is the microfluidics, because the microfluidics is important to, as I said, to have a very controlled bioassay, deal with very tiny volumes, and you can do all automated, especially because we use this Quakes valve. So the idea is that this is a 2-layer PDMS chamber chips that you see here mounted. And so you have the blue layer, that is the fluid layer, where we're going to put the different, here, some different chemistries. So they're going to be the 8 channels that we're going to use. And the orange part on top is actually controlling the valve. So you see that the red control channel crosses the blue channels. And because we are dealing with a flexible material, as we're going to inject some liquid or some compressed air, we're going to collapse the underneath channels. That's a way to have a computer control with these pneumatic valves of everything that is happening on the chip. So we're ready. We have the transduction mechanism, the ISCAT, we have the chemistry, we have the microfluidics, and then we can do an actual bioassay. And so this is what you see here. This is a video where you see some ovarian cancer extracellular vesicle that are actually landing on the surface. Each of these dots is a single vesicles, some of them of a few tens of nanometers. And so what we can do is to locate them, associate the contrast to them. And what I told you before is that the contrast gives you information about the size because the signal is proportional to the volume. So you have the occurrences, in that case, several thousands of them, and you have also an information about the size of each of these events, each of these analytes binding on the surface. So you can do that over a large portion of each of the 8 channels of the chip, where you have done a specific chemistry. And so eventually what you end up having is a huge amount of information. And so information is kind of processed in the way you see it on the right graph. We're going to plot the incidences as a function of the contrast. Please note that the contrast is negative. So basically, the orange part is the low contrast. So this is a background. So everything is related to remaining roughness or whatever. So this we don't want because it doesn't provide information. It's not related to the biosensing. And then you can isolate the size, the range of contrast that you want to study, because it will be associated to the size of the analytes you're interested in. So you can really choose how wide is this window, where it is positioned within the contrast, and because that enables you to be selective in the information you extract. And then you can start from real samples. So they are coming from, so that was a collaboration with Harvard Medical School, in particular the groups of Akoli and Young-Sum Him. And what you can see is that, for instance, here we take secretion from one cell line, the cancer cell line, and we functionalize different chambers of the chip with different antibodies, and you can see the whole dynamics of the bindings of the extracellular vesicle as a function of time. So now if we plot for 5 hours, we can reconstruct kind of a fingerprinting of, let's say, the EVs that are being secreted by these cells against the different kind of biomarkers with one control, that is IgG1. Let's not forget that all this information contain the size I've mentioned several times. And that's interesting to see that, in fact, depending on the kind of antibodies, there will be different population of sizes for the extracellular vesicle. So there's just very rich information that comes with the signal. And of course, you can do that for several cell lines and eventually reconstruct a whole, let's say, fingerprinting of the EVs from these different cells. And eventually, you can even identify and discriminate what were cancer cells, what were benign cells. What I mentioned before is the fact that these iSCAT have been pushed lately really to the amazing levels in terms of sensitivity. In particular, the two inventors in their own way have been pushing, let's say, detection towards single protein detection. So for instance, in the case of Kukura, there's beautiful work where he's kind of doing kind of a mass photometry where he's able to retrieve the molecular mass of molecular weight of different kind of analytes. In the case of Sandokan, what you see on the right, there is really what is right now the world record in detection of single protein without fluorescence. And that's very important. So typically 10 kilodalton, about 2 nanometer in size, that can be detected directly from the elastic scattering. So I'd like to show you something that is actually quite preliminary, but it's very exciting for us, is the possibility to do a bioassay with a single protein. It's actually a quite big one. This is fibronectin. And I'm going to show you 3 situations. The first one is no antibody to capture at the surface of a lipidic bilayer. And so basically, there's not much to see in the video that has just been played. The second situation is that you have an antibody, but it's not a specific antibody, it's a control antibody. So there's a little bit of non-specific interaction, but basically not much happening either. But if you put the right antibodies against fibronectin, you're going to see these black and dark spots happening. Okay, so you can really do that and look at each specific binding of individual protein, binding to a specific antibody, and you're going to see these dark spots. So that means that the molecule is landing. Then there's a pause, no signal. The molecule remains bound to the antibody, and then it is kind of leaving the surface. Okay, and so what you can do is, for the free situation, no antibody, non-specific antibody, and specific antibody, retrieve these entire kind of curves on hundreds of different proteins, individual proteins, and clearly evidence the specificity of the interaction depending on the case. So I've been talking a lot. How much time do I have? Can I use like maybe 8 minutes or something? Neha?
Yes, Professor Quidant.
Yes.
Very
Please
good.
go ahead.
Please go ahead. We have time.
Very good. So I've shown you that this digital holography enables you to detect analytes. So, and what we do here is because the analyte has a given up scattering, we detect the scattering, and that gives us information about the presence of the analyte. But you can go a step further. So this is not about analyte. You're going to see this is using some man-made samples, but the idea is to detect chirality, so optical activity. So I'm sure I have no need to actually define chirality, but just to make sure that we're all on the same page. So you know that chirality is what tells that an object is non-superimposable to its mirror image. So hands are chiral, but in fact, a lot of our, let's say, biomolecules in general are chiral. And so what is amazing is that this simple geometrical feature actually influences a lot the physics, the chemistry, and the biochemistry. So for instance, how biomolecules would interact with other molecules. And there is this very sad and famous case of what happened in the 50s with one drug that is named thalidomide, where unfortunately one of the enantiomers was actually a sedative drug, but the other one was actually generating many birth defects. So usually when you want to detect chirality, so you would do that most of the time optically and via detection of two quantities, what is called CD, circular dichroism. And circular dichroism is related to the differential absorption for light circular polarized, left circular polarized and right circular polarized light. And what is called ORD, this is the second quantity, this is optical rotatory dispersion. And in that case, it's not related to absorption, it's related to the actual refractive index that are going to be seen by the left circular polarized and the right circular polarized. But these two quantities are actually highly connected, and for, let's say, those who know what I'm talking about, so they are related via, conjugated via the Kramers-Kronig relation. Typically, you would have a setup that would measure CD, or you would have another setup that would measure ORD. It's actually not so trivial to measure two of them at the same time, and they have some complementary information. There's no special information. Usually you detect on a given volume, actually quite large volume and with a high concentration, but you do not map, let's say, domains of chirality. And it's also very time consuming, usually measurement, because you're eventually measuring a very tiny quantity. So there have been a few efforts in the past in trying to do chiral microscopes. And doesn't pretend to be exhaustive. I'm just showing a few cases. So typically, most of the time, except for the first example, that would be a confocal technique where you would be scanning the focus by detecting directly, the circular dichroism, or you would detect, for instance, a photothermal signal, or even the second harmonic generation. So what I would like to do in the time remaining is showing that an offline, off-axis holographic microscope can provide you images of optical activity, give you both ORD and CD at the same time, and to do that with an optical resolution. So let me guide you through the setup. So that looks like the original setup I was showing you when describing holography. So we're going to have the object beam. So this is from the laser on the left. You would be incident with a linear polarization, go through the chiral sample. And in the case, the chiral sample, as I said, will not be chiral molecules. Not for now. I wish that would be the case, but more structures that have been fabricated in the clean room. So they are gold nanoparticles, actually plasmonic nanoparticles that form some chiral patterns. When the light linearly polarized will go through, because of the structural chirality or optical activity, there would be a modification of the polarization. So I would go from linear to elliptical, and they're going to be due to CD and ORD. So this is my first part. And the reference beam in that case is going to be using some specific optical elements. I'm going to first generate two linear polarization states that are orthogonal, use a quarter wave plate to transform them into light that is left circular polarized and right circular polarized. And these two guys are going to send them and recombine them with the object beam. And this will go to the digital sensor. So what this means is that eventually here we're going to form an hologram that they're going to be two types of interferences of two types of holograms. So it would be the right circular polarized are going to interact with the sample beam. The left circular polarized are going to interact with the sample beam, and they will do that with a different angle. And eventually, this is the things we're going to have. So this is the hologram we measure. The square, the darker square, you see actually arrays of these nanoparticles. So one with left-handed, the other one right-handed. And these are typically 50 micron big. I'm going to do Fourier transform and don't enter into the detail, but because of different angle with which I formed the hologram, I can separate, especially in the Fourier space, what is left and what is right. Then I do an inverse Fourier transform, and I can then extract the amplitude of the field for left and right, and the phase of the field for left and right. And you can very easily convince yourself that the CD is going to be given by the ratio of the amplitude, left over right, and the OID is going to be given by the difference of the phase between left and right. And so you see these images that are directly image of CDs and OIDs. So then from then on, we can, for instance, check the variation of the image, so doing spectroscopic imaging, changing the wavelength. And so you see here the maps for different wavelengths, but you have an entire spectrum that is actually reconstructed on the right. And we do that both for CD and OID. And so what you're going to see is that first for the racemic, so I forgot to mention the sample has 3 blocks in that case, left-handed, right-handed, and racemic, so they're equal proportion. And we can check basically the nice feature of the CD for left and right. And we can also check the racemic that is flat. And then we can do the same with ORD. And so in the first case, we compare with a commercial CD method to check the consistency of everything. In the case of the OID, we don't have this comparison, this benchmarking, but we can check that the results are fully consistent with each other. So typically, you expect a maximum in OID to correspond to a minimum of CD due to the conjugation I was mentioning before. And I'm coming to an end. I'm showing now what we can do in mapping, let's say, chirality variation. So here, this is just playing around. So encoding the letter L and R using these different patterns. So the background in that case is the racemic. Meanwhile, the R is made of right structure, right-handed and left-handed for the L. So again, you check consistency of CD and ORD. But what is more important is that we can map enantiometric excesses. So you see this domain in this first experiment. So you say all the way, you go from one NDNS to the other by changing the ratio and creating some domain. So progressively, you're going to switch from right to left using this progressive arrangement. And you're going to be able to map the different domains. But I would say even more powerful, I think, is what you see here, is that you maintain the level to a racemic mixture. So that means if you would measure that with a CD meter, you would just see 0 signal. But because you're able to resolve the domains, you're going to see. So what we do in that case, that we maintain 50 and we decrease the size of the domains. And we're able to actually, until almost the very last one, to resolve the different regions where you have a right or a left NDNS. And this is, of course, something you would not be able to do with a CD meter. So I'm getting to an end. I hope I've not been too long. So as a conclusion, just to mention that I've been showing you 2 configurations that can contribute to the future of biosensing. The first one that enables to deal with heterogeneous population and to extract some information about secretions from cells. We focused on extracellular vesicle, but I showed that it's actually possible to push that down to the single protein level. And the second part was more showing how you can extract some additional information like chirality and optical activity. And in fact, what I didn't mention, but these same techniques can be used in different ways also to map the temperature with very high sensitivity and resolution, but also to manipulate fluids and monitor, let's say, the fluid dynamics. And so if you're interested in, for instance, learning about these aspects, I invite you to check our webpage and to identify the corresponding works. And so this is for my concluding remarks. So I'll stop here.